And thanks to ARM for inviting us to speak today. And today I'll be giving just a short introduction after some forward-looking statements about Fibrocell Science and our technology. Quick investment highlights. So Fibrocell Science focuses on developing rare and pediatric and also other diseases based on the platform of the autologous fibroblast cell. So what I'll do is I'll quickly go over some of our lead indications. What we do is we're developing gene therapies in the field of skin diseases. So the fibroblast is actually quite well designed for creating a vehicle to bring these gene constructs to skin. So our lead indication is called recessive dystrophic epidermolysis bullosa, RDEB, which is a rare monogenic skin disease that results in a blistering condition that's highly devastating to the patient. And right now we're in co-development with Intrexon to bring a candidate to IND by mid-year, and then to bring that into the clinic by the end of the year. And I'll have another slide just to give you a little more information on this disease. While that is in development, we already have a second program that's coming up behind that, another ex vivo gene-modified fibroblast to treat linearscleroderma. And linearscleroderma is a condition in which the skin forms these hardened patches or linear bands of scars that contains excessive collagen 1 and 3. And when these transverse joints, or if they occur early in development, they can actually cause developmental issues. So what we want to do is create a construct that will break up that fibrosis and then allow development to happen normally, or to increase range of motion and reduce pain in the joint. And we'll be also developing that one with Intrexon as well. We're looking for an IND on that one in 2016. And then away from our gene therapy programs, we also have a BLA using just unmodified autologous fibroblasts. And we carry a phase 2 SBLA program right now, and it's enrolled, and we're continuing some additional enrollment, and I'll talk a little bit about the timeline on the next chart. That is in vocal cord scarring. So that's a condition in which the vocal folds of the cords themselves thicken from scarring and also have issues with tissue development. And this can occur through overuse. It can occur from trauma to the area. It can also occur due to just some idiopathic reason, age, and also surgery, or even if you have, cancer in the area and you have radiation treatment. These can all lead to vocal cord scarring, and we think we have a solution using our autologous fibroblasts to treat that area. So just looking at our development pipeline in a whole, our lead program from a timing standpoint is vocal cord scarring. That is in phase 2. We have a 20-patient, multi-centered, double-blind study that we're currently enrolling that has 22 patients enrolled. So we're over-enrolling just in case there's some attrition in the follow-up period with a 4-month primary endpoint. And we're targeting to have this through all the injection treatments and follow-up points by the end of the year, hoping to report that out sometime around then or early in Q1 of 16. Our lead therapy program, which I'll have one more slide on, discusses RDEB. It's currently preclinical animal studies now. We're hoping to have that done mid-year, as I mentioned, and then in the clinic in phase 1 at Stanford University in the second half of the year. And then linear scleroderma is in product development phase right now. So just a quick note, my last minute, on what RDEB is all about. So you can see some cartoons of skin cross-section here, and if you focus on those anchoring fibrils, which are those sort of arcing structures that are connecting the sort of keratinocyte and basal layer of the skin down to the lower dermis, those are actually called anchoring fibrils, and they're constructed of collagen 7. So what occurs in this disease is there's a mutation in a gene called COL7A1, which encodes collagen 7. And this is a monogenic disease because when there's a mutation in this, the patient does not properly produce the protein. And then you get this situation on the right where the layers of skin are actually not anchored and they float on top of one another. So even minor sort of lateral friction can create blistering all over the skin and body. So the life expectancy is about 30 years, very painful. It's got no real current therapies right now, just really wound treatments. With a high economic impact as well to the families. And as you can see in the epidemiology, it's ultra orphan. So we are creating this ex vivo gene-modified fibroblast, lentivirus transduced fibroblast that then has the full COL7A1 structure encoded into the cell, and then we'll deliver that locally to the patient. Okay? And just a quick look at one of our patients who we could potentially target and target those wounded areas that we'd like to help. Okay. Thank you very much.
Great. Thank you, John. Great, thanks for that introduction. So to dig in a little bit more, as you described, you're using autologous fibroblasts to essentially endogenously express proteins or be essentially internal protein factories. So tell us a little bit more about the process. Why is the fibroblast itself important? And, what are you doing? How do you use them? What's the transduction yield? Tell us a little bit more about the approach.
Sure. So we have an existing platform where we grow autologous fibroblasts through a FDA-approved BLA process. So essentially, we took this same process and then modified it for the transduction step. Now, to answer quickly about why fibroblasts are important, being this is a skin disease, and if you know about keratinocytes, they have high turnover. We really want to find a skin cell that we can actually target that will have some longevity, may have, and we'll find this out in our models, some in vivo sort of replication where daughter cells have this integrated Col7A construct, so we can get some longer protein expression. So what we do in the manufacturing process, and obviously I can't get into terrible detail, but what I can say is, in early passage in our, and this is a very low passage process, less than 3 to 4 passages, we can will transduce around the early passages, and then because it's integrative, we'll then expand that population from the cells. Now, these are autologous cells. We take them from skin samples. We grow them up using fairly standard techniques. But then we'll expand that modified cell up into hundreds of millions of cells, if not more, and then we'll cryopreserve those. The goal being that we can then take those out and treat the patient over a longer period of time.
Great. Excellent. That's really helpful. So you mentioned RDEB as a rare skin disorder, basically in which the patient's fibroblasts do not produce enough of the collagen 7. You're going to be filing an IND mid-year and begin phase studies this year. So tell us a little bit more about the clinical trial plans, because this is a unique indication to be studying with unique endpoints and requirements. Tell us a little bit about how you plan to move into the clinic initially.
Yeah, that's a good question because, especially in pediatric population, a disease that has a large pediatric number with it because of the age expectancy, but mainly because it's orphan. So what we're planning to do is in the sort of phase 1 portion of the first program, we will be testing in adults. The FDA typically, even in gene transfer trials, look for adults, but this will be a relatively low number trial. We're targeting 3 to 5, in that first phase 1. What we want to do in phase 1 is establish a biological precedent. So there's a lot of work in the literature, if you've researched RDEB, where you can simply take, do biopsies of treated areas, do histology, and using immunofluorescence with, like an anti-Col7A1, or Col7, and even using electron microscope analysis, you can actually physically see the anchoring fibrils in the basement membrane zone. Or if you, use anti-Col7, you can see them fluoresce in the membrane zone. The goal would be to, in a RDEB modified cell from those biopsies, see now that you have fluorescence in the membrane zone because you localized the Col7 to the proper area. That means you made functional collagen 7 as well. And make sure they're aligned properly using the EM analysis. So this is our primary endpoint for the early phase, just to establish the mode of activity and the biological endpoint. We will also be looking at some secondary endpoint scales and things like that, but also 3D photography or some other measures to start looking now at what does the wound healing rates look like, how well do they stay healed, because those are things we'll use in later phases, more and more efficacy endpoints.
Okay, excellent. And then, assuming things go well in the phase 3, how do you anticipate moving to pediatrics, and what do you see as the regulatory strategy here?
Yeah, that's a good question too. Now, what we plan to do in this is we'll obviously have to clear with the FDA. We already talked to the RAC committee at the NIH about this. Is can we start in this adult 3 to 5 patient population in a hybrid phase 1 and 2, and then move into a pediatric population? Because unfortunately, because of this disease, there just aren't a lot of adults around. Now, luckily, we work with Stanford University, and they have a registry of patients that we will be using to to help with our recruitment efforts. But we would hope that somewhere we can come to agreement where we can show some safety analysis, obviously with follow-up, long-term follow-ups will occur in any gene transfer trial, but move into this population after we establish proof of concept start working at some pediatric numbers, and then expand that into our later phase studies. So that'll be the strategy. But of course, we have to vet that with the FDA first.
And what do you envision as the ultimate efficacy endpoints? Is it wound healing? Is it prevention of wound?
Right.
Is it quality of life? Is it some composite of these? Are you going to be kind of assessing sort of where you have effect as you go into phase 2 to really discuss with the FDA what makes the most sense for?
Yeah, and I think you're right. It'll be some sort of composite, but what we've been focused on initially, even early discussions with FDA, and of course working with our KOLs, are not only wound healing rates, but also length of wound closure, how long they stay closed. Because really the problem here is rewounding, causing these wounds to have this rewounding cycle. So we have some ideas of how to do that. We've already did some preliminary discussions with our experts and with FDA, but we'll sort of do that as a secondary endpoint so we can vet that and show that in later trials. But there are other mechanisms we can use as well if we have to come to some different agreement, things like torsion tests and things like that. But it'll be something along those lines, something about that wounding and rewounding.
And I'm going to move on to linearscleroderma.
Sure.
And here is a different collagen loaded into the fibroblast. And I probably should have asked this earlier, but how are you actually delivering the fibroblasts? Are they engrafting, and that's how they're presenting or expressing protein?
Yeah, this is actually the opposite approach from RDEB for linear scleroderma. So if you think about the condition here, you have this overabundance of collagen 1 and 3. And the goal will be here is to break up that sclerosis and then release that scar. So what we're going to do here is a little different. So we know the mode of activity of RDEB is pretty straightforward. It's a monogenic Col7 disorder. Not so much with linear scleroderma. You're dealing with autoimmune disease that can have several factors, environmental, what have you, that cause it. So here we're doing more of the sequelae of disease. We're going to treat that sclerosis. So what we're going to have here is some type of, and we haven't finalized the gene yet, but it'll be a gene, an interstitial collagenase or some sort of gene that will cause that breakdown. But if you're familiar with Intrexon's technology, we are going to fit this with the RIA switch technology. Simply, it's an inducible promoter system that has a ligand-based compound that the cells, when they're injected, will be dormant until you present them to the promoter region, this ligand compound, and then it causes it to turn on, and then it starts transcription. So the reason we want to do that in this candidate is once you break up the sclerosis, you don't want to keep constitutively breaking it up because your ECM will just start collapsing upon itself. So we're going to add that safety construct.
So it's really dialing in some control.
Right. It's some control, right.
Maybe just finishing up with kind of, you brought up Intrexon, the RIA switch technology. What really else are they bringing to the table, and how are they helping Fibrocell develop these advanced cell therapies?
Yeah, absolutely. So our partnership with Intrexon is it's obviously our main driver in our pipeline. So they essentially act as our R&D. First of all, they act as our R&D division. We have access to wonderful scientists, and they have an array of services that were originally, if we were trying to do this on his own, we may have to go to 4 or 5 different either CMOs or different providers to create the same contract. So it's one-stop shop. Not only do you get molecular engineering, you get cellular engineering, and they also actually have quite good regulatory oversight as well. Things like pre-IND meetings and IND packages. We're all working together with Intrexon on this. So that's the way we look at it, is not only do they obviously give clout to our company pipeline, they provide all these services under one roof, and it's actually an excellent collaboration for us.
Great, and I think we have just 1 minute left. The vocal cord program is a non-modified program. You said we may be getting data next year. How is that mechanism different?
Yeah, it's in gene therapy you can dial in the mechanism a little better, and the mechanism on that is a little bit more around. It's a cell therapy type mechanism. You always have some parts of it there that you're evaluating your clinical trials. But in this case, where you have scarring occurring, the fibroblast cells not only can produce collagen, but they also produce collagenases to break up scarring. So what we believe is happening here, and we'll obviously have to work to this, is that it breaks up the scarring and then turned over to remodel the scar in order to sort of regenerate the area. It's more in the regenerative medicine side. The goal there is the scarring sort of diminishes the frequency that the wavelengths that the folds can vibrate out, which diminishes voice. It's a condition called dysphonia. If we can recreate that, then we can recreate the wavelength of the scar, and we can restore voice.
Great. Excellent. Well, John, thank you so much for being here and providing such a good, albeit quick overview on Fibrocell. I appreciate you getting through in a short period of time, but thanks for being with us.
Thank you very much.